Hi, everyone. We're going to begin. All right. Good evening, everyone. I am Dr. Diana Lee, Associate Director of Education and Training Initiatives here at Columbia University's Zuckerman Institute. And it is my great pleasure to welcome you all to the third event in this year's Stavros Niarchos Foundation Brain Insight Lecture Series. So whether you're attending in person here at the Zuckerman Institute in our Kavli Auditorium, or if you're tuning in online, I'm really glad you're here for our lecture titled The Circuits That Move Us. So the Zuckerman Institute brings together researchers from departments across the university to transform our understanding of the brain and mind. And we're extremely pleased to continue sharing the incredible research through not only this lecture series, but through all of our free public engagement programs at the Institute, engaging the youngest of learners through high school students, through our most experienced adults. And we remain really dedicated to communicating the innovative, groundbreaking science in a way that is relevant, informative, and empowering for all of our community members. So we hope that tonight's presentation will invite you to explore the science happening at the Zuckerman Institute, but also across Columbia's campuses and New York City at large. And I'm hoping you'll be able to think about the many ways that science shows up in all of our daily lives. So this lecture series is paired closely with another one of our programs. Some of you may already know this if you've been here before. We have the Stavros Niarchos Foundation Teacher Scholar Program as well. And that's where a cohort of 24 amazing New York City secondary school teachers come together to create new ways to bring science featured in these lectures into their classrooms. So teachers who are in our audience and tuning in online, I'd like to thank you for your dedication and your commitment to making learning fun and inspiring that spark in your students. So these programs are made possible by the Stavros Niarchos Foundation. And I'm always so grateful to express my, grateful and gratitude, a lot of gratitude in this sentence to the foundation and its members who join us tonight. And I just want to thank you all for that generous, sustained support. It means so much to know we have that support to make these programs continue and to maintain this partnership where together we can make brain science accessible to all. And I think we can agree that this work remains really important, especially these days. So tonight's topic, we're all gathered here to talk about, no surprise, the brain, but I also like to think it's about community. And I'm sure you've been seeing collective action arising across our country in recent weeks, and I find myself reflecting on the question, what can we accomplish together that we can't when we are alone? And if we think of the brain like this, it's 100 billion cells. And when the right combinations of those cells activate at the right time, those cells can produce an astounding range of behaviors, and we can produce an astounding range of motions. So how does the activity of individual brain cells give rise to that behavior? And what happens when that precise orchestration falls apart? So in tonight's event, we have 3 experts who come together to address such questions, bringing perspectives from computational, experimental, and clinical approaches in neuroscience to see how form meets function in our brains. So we'll soon hear from 2 exceptional speakers, followed by a conversation between them led by Dr. Leslie Seibner. And Dr. Seibner will be our host and moderator this evening. She is a postdoctoral fellow in Dr. Priya Rajasethupathy's lab at Rockefeller University. She earned her PhD in neurobiology and behavior at Columbia University here at the Zuckerman Institute with Dr. Rui Costa, and her bachelor degree in neuroscience and creative writing from Johns Hopkins University. During her PhD, Dr. Seibner investigated the neural mechanisms underlying the learning of skilled motor action. Her work focused on how the thalamus, an often overlooked motor area of the brain, contributes to the learning and refinement of reaching actions in mice. Her postdoctoral research now examines how the saliency of events is reported to memory centers of the brain and why specific memories are selected for long-term storage while others aren't. She has been awarded with numerous awards, including a National Research Service Award from the National Institutes of Health and she's currently a junior fellow at the Simon Society of Fellows. She is really engaged in the broader scientific community, an advocate, and she works in science communication and education as well, with stories of women in neuroscience and BioBus, and she co-founded Scientists on the Subway. So, for those of you who are tuning in online, you have a Q&A button in the Zoom webinar. Feel free to use that to submit questions as you listen to the talks. And for those who are in person, when it's time for the Q&A, Dr. Seibner will invite you to raise your hand and ask your question into a microphone. So please join me in giving a warm welcome to our host and moderator, Dr. Leslie Seibner.
Okay, thank you so much, Dr. Lee, for that amazing introduction. I'm so excited to be back here at Columbia where I did my PhD. It's been a little while, but I'm just so excited to be here where we're going to be hearing from Dr. Leah Dunker and Dr. Hamali Fatnani, two experts who will be discussing how form meets function in the brain. Now, today's topic is one that personally really fascinates me. As Dr. Lee mentioned, I studied movement and motor systems during my PhD, and it's this huge question in the field of neuroscience that poses how can the brain actually work in concert to create behavior that actually allows us to affect the surrounding world around us. Now, this curiosity isn't just about the brain and how it works, but there's a balance that we can see in many different components of our daily lives. You know, we all probably have a cell phone that usually works exactly how we want it to, but when it overheats, that system will fail. Or on the flip side, you can have a tap where you get clean water every day, but if it gets too cold outside, like an historic cold front, a pipe might burst, and then all of a sudden, that system fails. So today, we're going to be examining a system when it is functioning properly, as well as when it is broken. So we're really going to hear about how the brain allows us to interact with the world with movement, and also learn about what happens when that motor system goes awry. First, we're going to hear from Dr. Dunker, and she will share her research that explores how the brain performs computations that underlie everyday actions. Then we'll hear from Dr. Fatnani, where she will discuss the significance of cell-to-cell connections in the brain through the lens of motor neuron disease, about the motor neuron disease, ALS. Each of their talks will be about 15 minutes long. Then I will moderate a discussion in which we will include you, the audience, as well as the online audience, where you can submit questions online. So for those of you who have already submitted questions, thank you so much. And for those of you on the live stream, remember to use the Q&A button to submit the questions to the speakers as they come up, and we will follow up with them during the discussion. When you are asking your question in the live audience, let us know if you're a teacher or a student. And if you're a student, please tell us what grade you're in. So now it is my pleasure to introduce you to our first speaker, Dr. Leah Dunker. Dr. Dunker is an assistant professor here in the neuroscience department at Columbia University and a principal investigator at the Center for Theoretical Neuroscience in the Zuckerman Mind-Brain Behavior Institute. Her research merges theoretical and data-driven approaches, developing computational models of neural networks and applying machine learning methods to analyze large-scale brain recordings. By integrating these perspectives, Dr. Dunker aims to uncover how neural circuits perform complex computations underlying cognition and motor control. Before joining us here at Columbia, she was a postdoctoral researcher at Stanford University, working with doctors Krishna Shenoy and Scott Linderman. She completed her PhD at the Gatsby Computational Neuroscience Unit at the University College London, working with Manish Sahani, and she holds an undergraduate and master's degrees from UCL in natural sciences and computational statistics and machine learning. Please join me in welcoming Dr. Dunker to the stage.
All right, thank you very much. It's great to be here. So I think in everyday life, it's sometimes easy to forget and really appreciate how amazing our motor system really is, right? So a human body can learn to scale up mountains. we can perform gymnastic routines, and we can learn to play a musical instrument, not just to create sound, but actually to convey some sort of emotional meaning and touch people with what we create, right? But when you think about it, even actions that we perform every day, like typing on our smartphone to send a text message to our friend, or reaching to grab a cup of coffee and take a sip, actually are pretty complex movements. Right, and underlying these movements are neural circuits that have evolved to perform computations. So the kind of computations that you might think about when studying the motor systems are, for instance, planning motor sequences under uncertainty and pretty serious risks, performing and executing precise and very accurate movements with very narrow error margin, responding to tactile feedback from feedback from the senses, so like tactile feedback or auditory feedback, and flexibly responding to it, or precisely timing and coordinating multiple effectors, or anticipating something about the weight or shape of an object before we even perform the action to reach for it. Okay. So there are really a whole variety of very complex and interesting computations that our motor system is able to perform seamlessly. And they all contribute to sort of, our natural motor behavior. And so in studying this, we really think that there's many different areas of the brain and networks of neurons that are that participate and work together to implement these kind of computations, and ultimately send signals to the spinal cord and our muscles so that we can perform the everyday movements that we typically take for granted. And so I'm a computational neuroscientist. So in my work, I'm really interested in the question of how networks in the brain actually implement and perform these computations that are underlying motor control. And so I typically approach this from really a basic science standpoint. But ultimately, I think having a more fundamental understanding of how the system works and functions can also really help us in providing insights into disorders of the motor system when these computations break apart. And there's a really inspiring line of work, I think, that sort of builds on this, where brain-machine interfaces can use a better understanding of how neurons communicate and talk to each other to actually pick up on those signals and even restore a loss of function. Okay. But okay, so how do we approach this question of how neurons perform computations? So I would love to study mountain climbers or gymnasts, but typically we actually try to break this down into much simpler behaviors, like just considering an arm reach, for instance. And this is useful because it helps us to simplify the problem while kind of still maintaining aspects of the problem that we find interesting, like studying motor planning or execution, maybe looking for sources of variability and why we can't perform the same movement twice, or what has to happen in the brain to facilitate learning new movements, for instance. Okay, so this will involve defining some kind of experimental task paradigm, which will involve an instruction, so say the direction and timing of a movement. And ultimately, the goal or solving the task would mean that we follow that instruction and perform the required movement. Okay? And of course, the system that implements and solves this task is our brain. And so this is really what we care about modeling, networks in the brain that solve these task-relevant computations. And so the kind of approach that we often take as theorists is to build abstract models of this. So I thought it could be kind of fun to actually do that together. And so if we wanted to model a network of just two neurons in the brain, then I would typically draw something like this. So we have two nodes in a network, neuron 1 and neuron 2. And they both receive some kind of inputs that relate to the direction and the timing of the movements or the task instructions. And we think they process this somehow and work together to produce an output, which maybe relates to the kind of spinal activation or the muscle contraction, something like that. Okay? And so we think that the action in this really happens in how these neurons communicate with each other to take this input sequence and transform it into an output sequence. Okay, and typically, the kind of measurement of a neuron that we care about are firing rates, so how active is a neuron at a given time point. Okay, so if we think about this for neuron 1, for instance, its firing rate might look like this. And if we look at this diagram in trying to describe the activity of the neuron, what we can really think about is that it should be composed of some combination of the drive of the neuron itself and what its neighbor in the network says, and then also some drive from this input that it receives. Okay? And so if we take these components and we actually just give them some names in our diagram, we can use them, we can transform this intuition into a mathematical expression where we describe how the activity of a neuron slightly in the future depends on its own activity, what its neighbor says, and the input. Okay, so we've just written down an equation describing how a neuron in this network behaves. And we can do the same thing for the second neuron with a slightly different equation. And we can keep playing this game, adding more and more neurons to actually come up with an equation that models an entire network of hundreds of neurons. Okay, and we can follow similar logic to write this down as one equation. We're kind of collecting the activity across all neurons in what is called a vector. And we're collecting the connection strengths between neurons in a matrix called W. And so this is sort of the most central equation to what I do, is thinking about this sort of like network dynamics. Okay, and this is interesting because what it helps us to do is to actually form theoretical hypotheses about how a network functions, right? How do properties of the connectivity of the network shape the sort of computations that it can perform? And how does that in turn shape the sort of activation patterns or, that a neuron expresses in the network? And of course, if we have theoretical ideas of this, then this will also guide how we look at neural data, right? How can we interpret the kind of activity patterns that we see experimentally? It might also help us to come up with ideas of how we should go about building methods for data analysis that could kind of lead to a similar description. Okay, and so I thought I would give you a little bit of an idea of how we apply these kind of concepts in our research. And I think a very fundamental question that we're all interested in as neuroscientists are really how changes in activity in networks in areas of the brain that we think are involved in motor control, how do changes in activity drive changes in the actual motor behavior, right? So very fundamental. Typically people approach this by taking an actual network in the biological brain and recording neural activity as an animal is performing a particular movement. So what I'm showing you here is an example of what this looks like from a paper that was published by Mark Churchland, who's actually a principal investigator in the building as well. And so what I'm showing you here is the firing rate of a neuron in spikes per second for many different arm reaches with different curvatures. This is the colors as a function of time, and this is averaged over many repetitions of the same behavior. Okay, this is what one neuron looks like. We can also measure many hundreds of neurons and apply techniques which are called techniques for dimensionality reduction, which allow us to actually visualize what these hundreds of neurons are doing in just a two-dimensional plot. This is what I'm showing you here. And here each gray line is showing how the population activity evolves over time as the monkey performs an arm reach. Okay? But the main takeaway point here is really that movement seems to involve structured and coordinated activity patterns at the level of networks of neurons in the brain. And these structured activity patterns are precisely thought to be implemented through these network dynamics that we already talked about. Okay, but a lot of this work so far is really just asking an animal to perform a behavior and measuring some activity in the brain, sort of trying to correlate those two signals. It's not really a causal test of what would a change in activity do to a change in behavior. Okay, so I've collaborated with an experimental group on this, where we thought that perhaps taking our theoretical understanding of how the system works, we could try to push neural activity around by adding a perturbation. Okay, so we can actually try to change activity and then see what happens to behavior. And there's two main ways that people have approached this in neuroscience using different techniques. One technique is called optogenetic stimulation. This actually allows us to change the properties of the cell to make it sensitive to light. So we can point a laser at the cell and it will start to fire, which is pretty cool. A second approach is called electrical stimulation, which does the same thing, but using an electrode to stimulate a neuron. When we do this in the brain, we can actually drive large changes in activity, which is what I'm showing you here. We're showing you a bunch of different neurons, the firing rates or the change in firing rates of a bunch of different neurons over time. And you can clearly see that when we turn on the optogenetic stimulation in this window, neurons start to fire. We can do the same thing for the electrical stimulation, and we again see that we can drive large changes in activity. what was interesting to us about this is that even though these two modalities seem to drive large changes in neural activity, it actually didn't result in a change in behavior in the optogenetic stimulation case. So what I'm showing you here are the reach trajectories of behavior. They look exactly the same between stimulation and without stimulation. Whereas for electrical stimulation, this change in activity actually created a change in behavior. Okay, so what is different between these two different approaches, and what actually changed about the activity patterns? Okay, so what we found was that actually only very specific perturbations to the activity also drive perturbations to the behavior. Okay, and so this really meant that very few patterns of coordinated changes in neural activity can drive behavior relative to sort of the vast number of neurons that are actually in the brain. And another thing that we really like to think about are high-dimensional coordinate spaces, but for today, we'll just work with 3 dimensions, where you can imagine all the possible combinations of activity patterns that 3 neurons can express as a 3-dimensional space, right? This is kind of like an x, y, z coordinate. You can go anywhere in this space. And what few activity patterns relative to the total number of neurons really means is that the only places that you can go to change behavior are actually just a 2-dimensional sheet. So you can kind of imagine me holding up a piece of paper. Okay, and so there's many ways that you could change neural firing off of that sheet of paper without creating an effect. And actually only a few ways you could point along the sheet of paper to change behavior. Okay, and this is actually exactly what we found when we looked at the experimental data and tried to interpret it through that lens, where we see that perturbations that changed activity along the sheet a lot when also created a larger effect on the behavior. Okay. So this was interesting to us, and especially so observing this in light of also thinking about the theoretical properties actually also helps us to develop constraints on what are valid hypotheses for these kind of dynamics that are still consistent with these experimental observations that we've made. And ultimately, this kind of work helps us to provide a tighter link between neural activity, properties of the circuit that actually generates this neural activity, and the kind of behavior that the neural activity creates. Okay? Going a lot faster than I thought. But so I've basically given you a little crash course on the way that we think about computational models in motor control. So this equation that I came up with is also sometimes called a recurrent neural network, and it's really at the core of a lot of computational neuroscience work. And I've kind of given you a little bit of an idea of how we use these concepts in thinking about causal links between neural activity and behavior. But in my group at Columbia, we actually also use this kind of framework to study a lot of different topics. So for example, an area that I'm very interested in is also, a question I'm interested in is also how we're able to sequentially, throughout different experiences in life, can actually learn different motor skills, and how do we then use those skills, the repertoire of skills that we have, and flexibly express it in new situations. This is actually a difficult problem when we study it at the level of neural networks. We also build methods for neural data analysis to see if there's these network level descriptions, if they're interesting, how do we build better methods to take neural recordings and extract these kind of descriptions from them. And generally, we're also very interested in questions around flexibility. So how do networks in the brain work together differently in different tasks to do different things, and what instantiates this kind of flexibility. Okay, so these are some research directions that we're excited about. Here's the people who I work with to address those kind of questions. This was us doing warmer days earlier last year. And yeah, thank you so much for your attention. It's great to be here, and look forward to the Q&A. Yeah.
Awesome. Thank you so much, Leah, for that amazing talk. I know that I have a lot of questions, but I'll have to hold on to them until a little bit later. And this is just a reminder for the folks watching online, while all of this is fresh in your brain, please use the Q&A button. to submit questions for the discussion later. And now it's my great pleasure to introduce our second speaker, Dr. Himali Fatnani. Dr. Fatnani is an assistant professor of neurological sciences in the Department of Neurology in the Division of Neuromuscular Medicine, a member of the Eleanor and Lou Gehrig ALS Center, a CTNI scholar at the Columbia Translational Neuroscience Initiative, and a member of the Center of Motor Neuron Biology and Disease at the Vagelos College of Physicians and Surgeons at Columbia University. She also directs the Center for Genomics of Neurodegenerative Diseases at the New York Genome Center. Dr. Fatnani's research program bridges Columbia University and the New York Genome Center and has three main goals: to be the hub of collaborative interactions between clinicians and basic scientists, to build and disseminate resources for the research community, and lastly, to understand the role of intercellular interactions in neurodegenerative disease. Her lab takes a multidisciplinary approach by leveraging clinical, genomic, computational, and imaging data to learn how communities of cells in the brain and spinal cord change with aging and how this causes disease. Dr. Fatnani is the recipient of the NIH Director's Transformative Research Award, and she was recently awarded Columbia's Herald and Golden Research Award in Clinical Sciences. Please join me in welcoming Dr. Fatnani to the stage.
Thank you very much, Leslie, for that wonderful and overwhelming, slightly, introduction. I'm very happy to be here. I thought this was a very interesting take on how to give seminars, which is why I said yes when they asked me. And it actually was a lot of fun preparing for this talk, because it allowed me to take a step back and think about how I would put about a dozen years of work into 15 minutes to give you guys a flavor of how it took us a village to understand what this village of neural cells does. So, without further ado, today we're going to talk about what happens when things go wrong in the circuits that move us. So there's three key concepts that I want you to take away about how we study or how we think about neurodegeneration. They're the cells of the CNS, the central nervous system. They exist in neighborhoods and communities, just like neighborhoods and communities in Manhattan. The second concept is one of selective vulnerability. Not every cell is going to get sick in the same way. And the third concept is pathology. When you look at brain slices from people's brains who have died of the disease, you see glopped up pieces of protein and debris everywhere. And these patterns are different depending on the disease you look at. So what's up with all that? That's what we're going to try to figure out today. So what are the building blocks of the brain? Well, they're the neurons that are responsible for receiving and transmitting information, as Leah just described. You can add to that cells called oligodendrocytes that provide insulation and metabolic support to these neural cells. Then there's these pink guys, the resident immune cells of the nervous system that help to survey the environment and fight off disease, or repair little broken bits of neurons, or chew up little bits of debris. Then there's these star-shaped astrocytes that help to nourish the cells around them and help to modulate how synaptic transmission occurs, how neuron-to-neuron communication occurs. Then you have the blood vessels that provide a link to the outside and help to meet metabolic demand. When you put all of that together, you see how intricate this ecosystem is and how many things need to work together in concert to help the brain to function. Now, what happens when it gets sick? This is a picture from a brain that has Alzheimer's disease, and you can see these purple bits are one kind of the pathology that you see associated with Alzheimer's. And they're not the purple bits are not in every green cell. They're only in some. And we don't understand the significance of that yet. And then there's another kind of pathology, the white stuff. That's the amyloid that you see in AD brains, Alzheimer's disease brains. Now, the white stuff and the purple stuff have different patterns. So there's a regional difference in how the pathology manifests as well. Now, we're going to look at all of these concepts in the context of the amyotrophic lateral sclerosis frontotemporal dementia clinical spectrum. And what this is a disease of the motor system, this stuff here, the motor neurons that arise in the brain and send signals down to cells in the cord, and the cells in the spinal cord that send signals to the muscles and cause them to move. The same cells that Leah is studying the computation of. In ALS, amyotrophic, you lose the connection between these cells and the muscle. So the muscles don't get this trophic or growth support, and they begin to waste away. So that's the amyotrophic part. When you look at the spinal cord, a cross-section of the spinal cord on autopsy, you see two white spaces on either side of this cross-section of the cord. So this white stuff on either side of the cross-section is the debris from the dying cells, the dying projections of the cells that come from the top. So that's the lateral sclerosis, white stuff on the sides of the spinal cord. And that's where you get amyotrophic lateral sclerosis from. It's a combination of the clinical description and the pathological description, both of which together you need to make that diagnosis. Now, what's underappreciated about ALS, particularly in North America, is that about half of the patients that present with these motor symptoms are also going to have some level of cognitive or behavioral deficits. So it's not just this bit of the brain and the cord that are messed up, it's these bits that can also be dysfunctional in ALS. And we don't understand why. In about 15% so this you can see in about half of the patients that present with motor phenotypes, right? In about 15% of these cases, they're going to meet the clinical threshold for a diagnosis of frontotemporal dementia, the second most common dementia after Alzheimer's. So can ALS teach us something about FTD, frontotemporal dementia? And can this whole system help us understand the relationship between selective vulnerability, regional differences, communities of cells, and pathology? So ALS has its own unique pathology. It shares this in common with the other diseases, but in a slightly different way. So in ALS, there's a protein called TDP43 that tends to glop up, just like the purple and the white stuff that we saw in AD. Now, all you need to take away from this is that you see all of these pictures, there's different patterns. And it looks like this glopped up stuff is in different types of cells. So there's different patterns of this glopped up stuff, and it looks like it's in different types of cells. That's all you need to know. In fact, that's all we do know. We don't know how this relates to the disease. We think we do, but we haven't systematically assessed that. We don't know if it's a clue or just a red herring, or maybe a canary in the coal mine. And that's what we're trying to figure out. The selective vulnerability in ALS comes from these green bits. So this is a cross-section of the mouse spinal cord. Blue is all of the cells that are in the cord. Red are all of the neurons, the cell bodies of the neurons that are in the cord. Green are the motor neurons. It's only some of these green cells that are going to go, and we don't understand why. So we want to understand how all of these different cells interact as the disease progresses, and what the dynamics of this are across both time and space. So The three key concepts: selective vulnerability, cells of the CNS, neighborhoods and communities, and pathology. How do they relate to one another in the context of ALS FTD? How do A and C, how does the pathology and the neighborhood affect this selective vulnerability? And what does the pathology have to do with any of it? How does it relate to what goes wrong? And how does that relate to how the disease presents itself in people? So what we study in my lab is how do you use tissues from patients to understand the relationship between how the disease presents itself and its time course, and what might be different in different patients. What does it relate to at the level of the tissue? And in cases where you have mutations that are associated with this disease, how do those mutations affect these observations? Now, needless to say, this is very, very complicated, right? How can you do this for a chronic, late onset neurodegenerative disease? Because the patients whose tissues have been donated for this study have passed away from the disease. So they've lived with the disease for varying numbers of years, and there are different stages of the disease process almost. So we need to know all of that information. So these post-mortem samples, they need to be collected with due care and attention. They need to be able to be subjected to the kinds of sophisticated molecular biology analyses that we're going to subject them to. And they need to be pathologically really well characterized. So the clinic has to take very careful notes. The pathologist has to take very careful notes. And the people who are dissecting the tissues for pathology have to be very, very careful in how they do it. Can you begin to understand the ecosystem that it takes to even begin to collect the samples for this study? How many people need to work together in a community to make this happen? And then once you have that, how do you decide which tech you want to use to collect the data? And once you have that, how do you make sense of all that information? So this is the bit that took us almost 10 years. So we put together, because this is particularly difficult for ALS, which is a very rare disease. So you need to collect samples from many different places. So at the Genome Center, we built this consortium that partners like 45 different institutions, all of whom contributed samples from patients. We have clinical data. We have their whole genome sequenced for all of these patients. We started small, but this grew into the largest such study in the United States. This also helped us partner with lots of different brain banks who donated tissues for these studies. And now we can relate the sequence of the DNA to the genes that are expressed through methods called RNA sequencing. So RNA is just which genes are expressed. DNA is instructions for all of the genes in the body. So which genes are expressed in the central nervous system. And then in parallel, we also look at animal models of the disease, and we look at cells by themselves, and we look at tissues from both mice and humans, and we subject them to all of these gene expression profiling methods to understand how the expression of these genes changes over space and time. So how can you measure gene expression in multiple cell types and locations at the same time? And here we use a method called spatially encoded RNA sequencing, which is just a method to do gene expression profiling without losing the information about the place on the tissue that the gene expression signal came from. And essentially, what we're doing is taking a slice of tissue. This is that cross-section of the spinal cord, remember? And you put it on a glass slide that has this array of spots on it that have these little pieces of DNA molecules that have little signals on them that are going to help capture the RNA from these slices of tissue. So then you put this tissue on the slide. This is a very sophisticated slide-on view. You poke holes in the tissue. You allow that RNA to diffuse out and be captured by these pieces of DNA on the slide. Then it's copied, clipped off, and sequenced. And then you read the sequence off in a machine. Now we're going to take a gamble with seeing if this video plays. It's proven to be very temperamental. Nope. So this method was developed by Joachim Lundeberg at the SciLife lab in Sweden. And this study was one of the first such studies that this method was deployed for at such a large scale. But conceptually, this is what it is. You can take any piece of tissue from any part of a plant, a human, or any animal really, and then take it out of the organism. Is it going to play now? Nope. Slice it into little bits. Put that slice on a slide. Here's the pink and blue stain that I was talking about. Here's an up-close and personal view of that. These are all the cells. These are the spots, the arrays on that slide with those little pieces of DNA. This is the side-on view. These are the holes. That's the RNA being captured by those pieces. Here's the RNA being copied. Lots and lots of copies. You clip it off. You put it in a machine that reads off that sequence. And then the computer maps those sequences back onto the tissue. And then you can put all of that information from all of those slices back to here look, for example, at how the green and the blue genes were expressed, where they were, and what happened. Now, that last little bit, to see where those things were expressed, actually takes a lot of very complicated math. And that was done in collaboration with a group at the Flatiron Institute called Rich Barneau's lab. And they developed this system that allows you to take lots of these measurements and make inferences about what's expressed where and how much of it, and how is it different in different cases. So we're going to make now use of that clinical heterogeneity, the fact that different patients are different, and we're going to combine that information with the pathology that we showed you by looking at different regions of the brain and their associated phenotypes. So the region of the brain that's responsible for executive function, the region of the brain that's responsible for language, region of the brain that's responsible for motor function, combine that information with the scoring for how much path there was, and then group the patients based on how the disease presented itself in those patients. So the ECAS is just an assessment of what deficits there were. So the gray are the controls. They don't have any disease. The yellow have one type of disease. The red have yellow plus other stuff, the cognitively impaired cases. And then we're going to combine lots of different methods to study gene expression, the spatial methods, plus we're going to take the tissue apart and look up close and personal at the individual types of cells. And then we're going to merge this information and see if we can make inferences about ALS states, and then we're going to use microscopy to validate some of that. We're also going to link that information to the pathology that we see. And then we're going to say, how is the structure affected? How is cellular function affected? And how are programs of gene expression across cells and conditions, how do they change? So when you do all that with that complicated math, we ask, does ALS cause changes to the cortex, other parts that are non-motor? And does ALS with no cognitive symptoms, is it the same as or different from the rest of it? So, long story short, no change in the number of cells, change in the function of some cells, change in some cells, but only in some regions of the brain. And when you have cognitive impairment, you have different changes. And these changes depend on where in the brain you are. So this is just a cross-section, and there's different layers. So when you have language impairment, you have stuff that changes all over. When you have executive impairment, you have stuff that only changes here, but only in this region. And the types of things that change, the types of functions that change, the cells that are affected are different in this case versus this case. That's all we need to know. So there's lots of nuanced changes to these communities and neighborhoods that are associated with specific types of disease on the ALS FTD spectrum. And we wouldn't have known that if we hadn't done this really complicated stuff. Now remember, we said we want to link it to the path to see if it's a clue or a red herring. To do that, you need to do some more microscopy. So remember those slices of tissue that we said we were going to collect? So we've done this bit. We've done all the gene expression part. But now we're going to intercalate this with different types of measurements. We're going to link this to where is the pathology, what does it look like, which types of cells is it in, and what are the cellular programs that are affected in those cells. And then we're going to interlace those types of measurements with the gene expression measurement. And we're going to do that in over 100 patients in all of the regions that we know are affected. So then we can say, is it the region, is it the patient, is it the circuit, what's going on? And we wouldn't be able to do any of that without some very talented people and the whole village that it took to do this, from Joachim's lab to Rich's group, and all of the people present and past in the lab, and all of the people who provided the funding, and most important of all, the patients who donated their tissues for this analysis. Thank you for your attention.
Okay, thank you so much, Himali, for that incredible talk. I'm going to invite Leah and Himali now to join me up here for a little bit of discussion about all of the very interesting topics that we heard about here tonight. And, you know, personally, this sort of event is what really gets me excited about neuroscience, because these are obviously two very different researchers who tackle the brain in very different ways. And so what I'm hoping to kick off this Q&A with, I will start the question and then we'll alternate between in-person questions and questions online. But just to kick us off, I'd love to hear from both of you to help us try to find some linkages between what both of you do and where we can find connections. So after seeing each other's talks, what is something that sort of we can inspire in your own work, or something that just makes you stop and think, how could I integrate this into what I'm doing?
Should I go first?
Sure, yes.
Well, I think seeing Amali's talk was really, there's so much complexity in the system that I just drew like notes and connection weights on the slide and was like, look, we're modeling a neural network. And there's a lot of richness and complexity in that question as well. But then I think seeing different cell types, even like glial cells, so we don't even typically include in neural network models of sort of how the system computes. And I think there's just like so many ways that you could zoom in and find richness and complexity in the problem. So I think incorporating some of that that's clearly very relevant for loss of function into network models describing function would be, I think, one contact point that like immediately, like slide one, stood out to me. Yeah.
And what I was struck by is, so we're noting that, remember I told you that it's in particular layers that you're seeing these changes, and some of the changes that you're seeing are implicating how neurons communicate with one another. So if the fundamental biology of how they communicate with one another is the same, why is it only selectively happening in certain regions? And are those connections different, and how? And how might you be able to take that information and see what the implications of that difference in connectivity are? So that's what I thought was would make this really tractable.
Yeah, absolutely. So maybe I'll start with a question in person, if anybody just wants to raise their hand, and we have a mic runner who will come down and get to you. So we have someone, maybe we'll start with the right here in the green sweatshirt.
Hi, I'm Zara. I'm one of the teachers. I was really interested in the RNA sequencing because I teach a lot about DNA sequencing. And so the idea that there's like the wells for it to drop into and then make a copy using DNA, does that mean that you've it's not like loose nucleotides, is it a copy of the genome that goes with that RNAs, and does every single well have the full genome, so it just attaches where it connects?
So no, it's just, so now they do it differently. Now you have probes for each gene, and it's just a spot. So the spot is where the RNA is captured, and then you're going to read out what are the sequences of RNA that were captured in that spot. So that's what allows you to say which genes are expressed in the cells in that spot, and where is that spot on the tissue.
Great. Sure, yes, we can take this one.
Thank you for both extraordinary lectures. I have an unfair question to ask of the First Lady. Is it possible to roll down the screen and can you explain the formula to us?
Oh, sort of the big one that I showed.
Yes.
Yeah, so you really try to collect. So we're interested in, so this was another thing actually, like we're like all about studying activity of neurons, and there's so much you can learn even from dead tissue. So you collect the activity of each neuron into sort of like a long object called a vector. So this is, you have n neurons, it's kind of like a list with n entries. And that is going to change over time. And so we're trying to describe how the activity of all these neurons changes over time as a function of its own activity, so the activity of the network, so what are all the neurons saying, and how is each neuron connected to every other neuron. That was this W matrix that I had. So this is the connection strength. And then a network typically receives some input from other areas or from sensory systems. So we tried to sort of lump all of this into another term that's this input that I had. But we really think that the activity that we measure is some combination of an input drive to the system and how much each neuron listens to all the other neurons in the network. And that's what that equation tried to express. Mm-hmm. Yeah.
Is the work of both scientists published somewhere that we can read it?
Yeah. There's also, the Zuckerman Institute has a website, so that's where my publications can be found. So yeah.
Great. Okay, so we have a question from online that I wanted to field to both of you. I feel like you can answer it in maybe different ways, but they were curious about if a motor neuron dies, or maybe also for Leah, if just a neuron in your network dies, does the circuit die? What actually happens in that instance?
So the system is surprisingly resilient. Actually, it takes a lot of neurons to die for the movement to be affected. And there are, so if one connection dies, others can sprout and take its place. And if one type of all of the connections from one type of neuron, like the fast firing ones go, then the slow ones come and try to take that place. So the system tries to compensate. Disease happens when that compensation fails, and when it fails enough times or sufficiently that you can see clinical symptoms. So by the time you actually see clinical symptoms, a lot of damage has happened.
Yeah.
So just like we saw in the ALS, in the non-motor parts of the ALS FTD brains, right? You can see that there are changes already happening in those regions of the brain, but there's no clinical symptoms yet.
Yeah. That's interesting, because I feel like what that just struck in my mind is this idea of this resiliency and compensation that occurs. And maybe, Leah, are there in models of neurodegeneration or just models in general, how much sort of compensation or resiliency of that is baked into what you have or what other people have done?
Yeah, I think that often having sort of network models of systems that can sort of adjust over time is actually quite difficult, because you have to really figure out how does that compensation happen, and how can function just be taken over by a different part of the network? How does it happen through some learning process? But knowing that a neural system can actually do that, and that maybe one neuron dying doesn't suddenly send your whole brain into chaos, is actually like a pretty good constraint or sort of inspiration for building these kind of networks. So questions around robustness to noise, robustness to cell loss, maybe some robustness to like a gradual degeneration, those are all things that we can sort of take as inspiration and then think about how do we translate that into a mathematical framework in a model, and then study that model and see if that matches sort of phenomena that we see in neurodegeneration. So I think this is often, it's sort of like a back and forth between having it in the model or finding inspiration and putting it in the model. Yeah.
That's great. Maybe we'll take this young person in the sweatshirt.
Hi, I'm a high school senior, and I was wondering if in your work you've tried to explore a neurochemical approach by using like MRS instead of solely microscopy when you're trying to figure out what the structural issue is.
If we try to use what? I'm sorry?
Like the chemical, like trying to find like a snapshot of like all the chemicals that are there, and if there's like a deficit in a specific chemical that you can't find through microscopy.
Yeah, so that's an excellent question. So when we're doing microscopy, you're limited by asking, is this different? Is that different? You can't profile in an unbiased way what is different. And so there are methods now to do that in a spatial way, to look at what metabolites are different, what lipids are different. And we're partnering with some people who are doing that on the same blocks of tissue that we do the RNA and the microscopy on. So they're going to take little sections that intercalate with that and look at exactly those kinds of questions. And then there's other groups that are looking at detailed mapping of the synaptomes, at how the connections between the nerve cells are different. So you can say one slice is going to go for gene expression, one for microscopy, one for mass spectrometry, one for synaptome mapping, and all of that is going to get grouped together. So we're going to maximize the amount of information that we can collect from one block of tissue. Very good question.
And then we can take this one right here.
So going back to what you said about it being difficult to diagnose physically until like a lot of neurons have like had an issue, and you also mentioned in your presentation that it may be something that like in Western medicine that we're just not quite seeing. I know in a lot of other countries, healthcare is approached cohesively. Would that make, I'm gonna give you like a lot of steps. Would that make a difference in like diagnosing things? Is it something that we're just limited in Western medicine? Like, are other countries doing a better job at catching this sooner? And then, would anything change if there was a way to ethically study the same thing in people that are live, like as it's happening currently instead of post-mortem?
So the first question, it isn't about how we approach it that's the problem. It's you don't get symptoms until a certain threshold is met. So for people who are following, there's a whole field where they're trying to see when does this phenoconversion happen? How early can you catch the disease? And what kinds of recordings, physiology recordings, electrophysiology recordings do you need to do to be able to catch that event as early as you can? Because some of the gene therapy modules, the treatments that are coming online, the earlier you give them, the better it is. So people are actively investigating that. There are also digital approaches, so people can take voice recordings and look for subtle signals within that to see if you can catch some of that stuff early. So there's lots of interesting research happening around that. And then remember what I was telling you about looking at which connections might be selectively affected for the synapses? If you could identify that, then you could identify imaging-based biomarkers. You could give them things that would light up those particular connections in live people and see if, but this is many years down the road. So a lot of stuff you have to know a lot of stuff before you can get there, but that's why it's so important to know that kind of stuff.
Okay, I'll take another one from the online submissions. So people are obviously here interested in our own general health and well-being. And so we have a question here that is wondering about what we can do to best preserve these circuits for longevity, and if there's a way to recover nerve function after something has gone awry.
I'm not really a doctor, not that kind of doctor. So take my advice with a huge dose of salt, but I would say that clean living and high thinking. Eat your fruits and vegetables, eat a varied, healthy diet, stay active. People have tried to look at connections between lifestyle and ALS, and there are certain kinds of demographics where certain groups are more susceptible, but we don't really understand those relationships very well. And certain kinds of dietary interventions have been explored, like to see if certain types of diet might work better, but there's no strong evidence for that either.
Maybe we can have with the glasses and the blue sweater right there.
Thank you. I had a question for Leah. It's kind of, I don't know if it's super answerable, but I'm a senior at undergrad, and I'm writing my thesis on like computational neuroscience. So we're modeling using the hierarchical drift diffusion model. And I was just wondering if you, like what kind of model you guys use to do your modeling essentially, and if you had experience working with an HDDM or just a DDM in general. Are you creating your own models? Like I'm just curious about like how you choose your models, because that was a really big part of my thesis.
Yeah.
So I'm just like, curious about the process.
Yeah, that sounds like a great project for a thesis. Yeah, I think it really depends on like there's different approaches towards modeling, and so like I think drift diffusion models also have like a rich history at Columbia and in this building, because Mike Shatland has also done very influential work, and thinking about decision making in sort of that context. Personally, I build up these network models and then really think about sort of the time evolution through the network, and that's also called a dynamical system. And there's particular analyses that can fall out of that framework that I typically try to sort of, if I study a computation of interest, like so think about how it could fit into that framework and what parts of the system can we analyze to understand how it would compute or perform that computation. I think there's some work that actually frames drift diffusion models in sort of that realm as well. So I think there's connections. But often I also try to not be so married to a particular type of approach. I think it's really like a lot in theoretical neuroscience. I think a lot is about figuring out the right question to ask and the right approach to answer it. And I think being sort of flexible and coming up with ideas. And I generally just sort of, yeah, I find that there's a lot of value in a diversity of approaches. Yeah, so I sort of avoided answering your question.
I think I saw some other hands a minute ago. Maybe here? Yeah.
When I saw the picture of the neural network, I kind of first immediately thought of Markov chains, and I know there's a link, but I don't know what it is. And it reminded me of the story of the Manhattan Project and how they used a Markov chain to map the chain reaction and fission. I was wondering if there's kind of a similar mode of thinking there with neurons, and if there was maybe a chain reaction in really complex movement, or even pushing it over to like consciousness and overthinking or something similar.
Yeah, this is great. So I sort of avoided using words like Markov chain on my slides, but it's exactly right that this idea of modeling the temporal evolution is actually kind of asking how does the state of the network now influence what it's doing in the future. And that's kind of also the idea behind Markov chains, that there's kind of like a state that tells you how likely is it to go somewhere in the future. And that's exactly sort of these ideas around computation and neural circuits, is that the activity really pushes itself forward. And the way that unfolds, so it depends on the connectivity of the network, but ultimately also reflects kind of a computational process that unfolds in time. So it's really kind of how that Markov chain evolves over time is a signature of the computation that the system performs. So it would, if you make a decision about like left or right, it would end up in a different location if you think left or if you think right. And this is also, we think about movement plans as a planning process that unfolds. The movement itself, like producing the activity that will move the arm in the right way, is also like something that unfolds in time. So it's really everything you can think of as like a Markov chain unrolling, and we're trying to really describe how that chain evolves. Are there equations? Are there predictions we can make about it? So yeah, this is a great question. Yeah.
Yeah, and that actually dovetails well into a question that we have online, where, Leah, you're talking about how in these individual movements, there's this catalyst that will prime you for the next and next movement. But there's a question online that's asking about, specific movements or plans. You talked about this diversity of movements that people can generate. Are there different circuits in the brain for different kinds of movements? Is that understood?
Yes, great questions. So there's definitely neurons that kind of, we often also talk about tuning in neuroscience. So there's neurons that will typically, or even areas of the brain that will and be more involved in hand movements and maybe like facial movements. So there's definitely aspects like that. But then between, like if I'm playing piano or typing on my laptop, like that both involves my fingers, but maybe it involves quite different cognitive processes, so whether I'm like using language or sort of read sheet music. And I don't think we really know exactly like what are the mechanisms that facilitate that flexibility to switch between writing an email and like playing an interesting piece on the piano. Or so that's also something I find super fascinating. Like how is it that our motor system can actually do that, and I can learn these different skills without overriding how to write my emails in the morning. That would be bad for my job. Yeah, I think those are great questions, and I think that's also what makes neuroscience exciting, is that we don't actually have all the answers about those things, and there's still room to be creative and to build models, and yeah.
Definitely. Maybe up here in the front row.
Hi, I had a question for Leah about there was a slide where you're showing like optogenetic versus electric stimulation, but it seemed like you were computationally modeling that. And I was wondering like, where does that signal come from? Like, is it somehow from like a real brain recording, or are there just like certain properties that are common to those kinds of stimulation?
Yeah, another great question. So I think I sort of reduced the models that we actually use a little bit too far. So sometimes we actually care about cell types, and especially in optogenetic stimulation, typically to make lights sort of responsive, cells responsive to light, you target particular cell types to say, well, there's two classes of excitatory and inhibitory cells in the system. Let's target all the excitatory neurons. And so we can include that in a network model and then try to understand a similar perturbation sort of at the level of a model, and then compare that to the perturbation that was actually administered in the experiment, to kind of try to connect those two approaches. Yeah, so that's kind of how we do it. But there's usually always a mix between analyzing data, coming up with theories about how to describe the data, and then going back and forth. Yeah.
Wow. This has been a great Q&A session, so thank you so much for all of your input. I'm just going to end with one last question for both of our panelists. I guess with all of these exciting ideas that we've discussed for your field, for your work, what would you love to see next? Big question.
I'd like to see those maps that I was telling you about which synapses are where, and could that explain why you see only certain parts of the circuits affected and only certain subtypes of neurons. Mm-hmm. And then also there's three interesting things for me. One is this map of connections. The second is how do these glial cells interact with the vasculature, and how does all of that relate to the pathology? So there's stuff about, is it a clue, a red herring, and a canary in the coal mine? That's what I'm most excited about, because that pathology you see across diseases. So why are different diseases different if it's the same stuff that's being glucked up? Mm-hmm. And how is it different in different parts of the brain?
Oh, thank you.
Yeah, I think maybe the complexity of the actual biological system that I also used in my response to the first question of the Q&A. I think that seeing all the richness and sort of genomic, transcriptomic information that we actually have in the system, figuring out how to incorporate that into models. And then maybe also like a second axis of complexity. I think sometimes to come up with tractable descriptions of a computation, we have to simplify it to the extent that I think there's more complex naturalistic computations, like sort of the feedback that you have to get when you climb and like a stone breaks loose and you have to rapidly change what you're doing and taking uncertainty into consideration. So I think pushing the task complexity in a controlled setting and sort of really studying more difficult computations, I think will also be super exciting. So maybe eventually I get to study gymnasts. So.
Yeah. Okay, so that's all we have time for our discussion. And before I turn it back over to Dr. Lee to close our event, I just want to thank everyone here and online for attending this event, and to our speakers for educating us so much. This is a reminder to please try and take 2 minutes just to fill out the survey that we can put in the chat of the Q&A, and let us know in person if you'd like about your thoughts about today's events. And I'll hand it back over to Dr. Lee.
All right. First of all, round of applause for our amazing speakers, the moderator. Okay. I just have 3 things to say. First of all, thank you to Dr. Dunker, Dr. Fadnani, and Dr. Seibner for your engaging talks and insightful discussion. Thank you all for coming. It is not warm out there, and I appreciate you trekking over here. If you enjoyed this, we have one more lecture in our series. Our 4th one will be Wednesday, April 22nd, featuring Dr. Mimi Shirasuhiza, Dr. Wes Gruber, and Dr. Amanda Gonzalez-Segarra. And with them, we will learn about what the fruit fly can teach us about our brains. So until then, please continue to be well and have a good night.